So, good evening. Uh, Welcome to today's session. So, I apologize for the canceling two classes. So, I think I'll have to take two extra lecture series in between to compensate for the classes, and I'll be letting you know the date and time when I'll be taking it. I'll share a poll request asking for whatever time is suitable for all of you, and then I'll be taking that extra two classes accordingly. So let's move to this today's lecture. So today we will be dealing with the C3 photosynthesis. So in the previous classes, we went through what is the overview of the leaf anatomy, the plant anatomy, plant from plant to leaf to the cells inside the leaf where the photosynthesis is occurring. And then we learned about what are the light reactions, dark reactions, photorespiration, how is it different for different kinds of plants. There is C3 photosynthesis, C4 photosynthesis, CAM photosynthesis, and what is the evolutionary history behind this evolution of these various photosynthetic parts. From this lecture onwards, that is today's lecture onwards, we'll be focusing only in C3, only on C3 photosynthesis, and going more deeply into C3 photosynthesis and how we will model C3 photosynthesis. So the first couple of classes will be modeling the leaf scale photosynthesis. We will not be modeling it for the entire plant, but we'll be learning about how do we model it for a single leaf. Now, from a single leaf, we can scale it up to, we can model for the entire plant. But now we'll be learning about how to model the C3 photosynthesis for a single leaf. Now, in your earlier classes, we talked about the photorespiration C3 photosynthesis, C4 CAM photosynthesis. So let's get into C3 photosynthesis. Again, just to revise, we had also gone through what is C3 photosynthesis. The photosynthesis in the C3 photosynthetic pathway, there are two parts to it, the light reaction and the Calvin-Benson cycle, or the light independent or dark reaction. In the light dependent reaction, the light and water is used to convert, is used to obtain chemical energy, and this is happening in the thylakoid membranes of the chloroplast. And as output, we get these compounds called NADPH, ATP, along with oxygen. And then these molecules are then again used in the Calvin-Benson cycle, which happens in the stroma of the chloroplast, to which helps in the manufacture of carbohydrates. And again, this is where the light reaction takes place. This is where the Calvin cycle takes place. And we also talked about photorespiration, which is the process in which instead of carbon dioxide, oxygen is RUBP combines with oxygen with the help of the Rubisco enzyme, and it goes through a series of cycles to waste the carbon dioxide, which can otherwise be fixed for the generation of carbohydrate molecules, and then uh, utilizes this carbon dioxide. So it's a waste process, as a result of which a lot of energy is lost during this process. And this is one major reason for the evolution of C4 photosynthesis. Now, today, what we are going to look at is another sort of respiration called the dark respiration. Now, this respiration is a respiratory process that is happening in plants, not as a waste. I mean, photorespiration is the process that is happening in the stroma part of it, stroma, peroxisome, and mitochondria, which is a wasteful process. But there is another type of respiration also called the dark respiration, which we also call it the growth or maintenance respiration, because this respiration is helping the plants for various activities that help in the growth of the plant, repairing various parts of the plant, and various other cellular activities. So dark respiration is referring to the process by which this glucose that is manufactured as a result of the Calvin-Benson cycle, it is breaking down this glucose to give us to give the plant energy. Now, this process is called dark respiration because this is not happening in the presence of light. Instead, it is happening in a plant all the time, irrespective of whether there is light or not. So, if the plant is alive and it is active, then this sort of respiration will take place. And this respiration occurs in the mitochondria of the plant cell, like how the photosynthesis and the photorespiration process is occurring in the chloroplast, inside the chloroplast, the thylakoid membrane and the stroma. This dark respiration is occurring in the mitochondria of plant cells. So here, when photosynthesis is using light energy to convert carbon dioxide and water into glucose and oxygen, dark respiration is using oxygen and glucose to produce carbon dioxide, water, and ATP. So, like I said, it is happening day and night as long as the plant is alive. And there are three major cycles to this dark respiration. First is the glycolysis, then is the citric acid cycle, and next is the electron transport chain. So we will not be going into the details of all the reactions that is happening in any of this, but this is just to familiarize with this dark respiration and to be able so that you're able to compartmentalize photorespiration and dark respiration and not get confused with them. Now, although this is not occurring as a result of light, it is indirectly related to photosynthesis because it is as a result of photosynthesis that we are getting glucose and oxygen. And that is what is being used during dark respiration to produce its byproduct. And the major advantage or major use of this dark respiration is how it is releasing energy for the growth, repair, and other cellular activities in a plant. Now, like I said, in C3 plants, right now we are only talking about C3 plants. Let's forget about all the other kinds of plants. We will only be dealing with C3 plants. So in C3 plants, this mitochondria is available in various parts of the plant. Like it is available in the palisade mesophyll cells, spongy mesophyll cells. It is available in bundle sheath cells. It is available in the guard cells of the stomata. It is available in the cells of the roots and also in meristematic cells, meaning the stem cells that the cells you can find at the tip of the shoot and the tip of the root. So here in a plant cell, this is say the palisade mesophyll cell, and within this you can see the chloroplast where the photosynthesis is happening, and inside here there is another organelle called the mitochondria where the dark respiration is taking place. Here you can also see this is this is the root structure, and in the root structure you can see the root is also having cells, and within the cells, although it is not having the chloroplast, it is having mitochondria where the dark respiration is taking place. So like I said, there are three major processes in dark respiration. First is the glycolysis. The glycolysis is occurring in the cytoplasm of the cell. So this cytoplasm that is outside the mitochondria, the glycolysis will occur. And this process of glycolysis is nothing but when the glucose molecule is broken into pyruvate molecule through a series of enzymatic reaction, then we say that the glycolysis process happens. Now, this glycolysis process is resulting in production of 2 ATP, 2 NADH, and 2 pyruvate molecule. Now, you have to be careful with respect to this energy molecule. It is not NADPH, it is NADH. So, as a result of glycolysis, the plant is gaining ATP, NADH, and 2 pyruvate molecule. This 2 pyruvate molecule is going to get converted into acetyl CoA, which is going to enter the mitochondria for the Krebs cycle or the citric acid cycle. Now, this acetyl CoA, it will go through a series of reaction and will release out carbon dioxide as a waste product. Like in photorespiration, although carbon dioxide is wasted without being fixed, this carbon dioxide is also being released out, which will either go out or it is utilized. It will go within the if it is within the leaf cells, then it is going to circulate within the spongy mesophyll for the process of carboxylation. And even during this process, energy molecules like NADH and FADH and ATP are produced. In the third process, all these these NADH and FADH, all these are electron carriers. These enter into the electron transport chain, and it is here in the electron transport chain a proton gradient is created, and uh, this is again releasing a bunch of ATP molecules, which is driving the ATP synthase, similar to what is happening in the light reaction. Although it is not a light reaction, ATP synthase is being driven by the proton gradient that is created as a result of this electron transport chain. As a result of it, around 34 ATP molecules per glucose molecule is released. So you can see here this is the place where this is the glycolysis process happening in the cytoplasm. Pyruvic acid is formed. Pyruvic acid is converted to acetyl CoA. Acetyl CoA is then entering into the citric acid cycle, and then it goes into the electron transport chain. In the citric acid cycle, as you can see, a lot of NADH is generated. And if you see this diagram here, it is giving a net gain of energy molecules. That is, in glycolysis process alone, 2 ATP is generated. During the Krebs cycle, 2 ATP generated. And during the electron transport chain, around 32 to 34 ATP is generated. So in total, around 36, 38 ATP molecules are generated. So this dark respiration is a very important process similar to the photosynthesis, which is happening in the mitochondria cells. Okay, so for now, what I'll do is I'll launch a small quiz which you can answer, and you can just submit. I'll wait for 5 to 10 minutes. It's a very simple quiz. So you can Okay, so you will be able to see a quiz in your on your screen. You can just answer those quiz, and I'll wait for 5 minutes. You can just do it and maybe put a note in the chat box so that I know when all of you are done. Are all of you able to view the quiz? Can someone just respond?
Yes, ma'am, the quiz is visible.
Okay, thank you, Shreyas.
Ma'am, the quiz has disappeared. Can you share once again?
Are you able to see it now, Shreyas? Okay, I think I'll end the quiz. Does anyone of you need more time? In that case, I'll keep it on. Otherwise, you can just, if you don't respond, I'll be just ending it. Okay, no problem. Yeah, sure, sure, sure. Manish, I think you just entered. I launch this quiz. I'll share it with you in the group so you can go through that during that time. Shreyas, are you done with the quiz? Okay, okay, okay. Okay, do me a favor, just drop in a message when you're done. So then I'll end it that time. Manish, were you saying something? Actually, a quiz is going on, Manish. So since you entered late, I don't know, maybe because of that you might not be able to see the screen. Um you can exit and rejoin once again. I'm not sure if that is the reason why you're not able to see the screen. Okay, Manish, don't worry, I'll be sharing this quiz and the solutions in the group or by email. So I think you can take your time and just practicing it on your own. I'll for now be ending it. So okay, I'll share the results later on in the group or by email. So let's move on to our next part of the lecture. So this is a portion till now we had looked at various processes that okay, Shreyas, no problem, that's okay. I mean, this is not being evaluated, it is just for your revision, so no issues. Okay. So till now we were going through all the theoretical portions or what are the reactions happening. I think pretty much all of you might be very familiar with all these processes since all of you are coming and most of you are coming from the botany background. And I hope all those of you who are not having that background have got an idea about the processes in focus and what we are going to be focusing on with respect to the photosynthesis process. Now, now we'll be entering what is required when we try to numerically model or we try to develop a model that will help us in obtaining this photosynthesis rate and uh, transpiration rate and all through equations. So one important uh, element required for this are the photosynthetic response curve. So photosynthetic response curves are nothing but they are describing how photosynthesis is varying or responding to environmental factors such as light intensity, intercellular carbon dioxide concentration, and temperature. This response curves are giving us what will be the rate of photosynthesis for varying values of carbon dioxide, for varying values of light, for varying values of temperature. And these curves from these curves, we are going to understand what are the factors that limit photosynthesis or increase photosynthesis or decrease photosynthesis. And from knowing these factors or the limitation factors, we are able to understand how efficiently that photosynthesis process is occurring in a plant, not for one condition, but for different conditions. So the three typical or three common type of response curves that we will be looking into and that we'll be using are the ACI curves, AQ curves, and AT curves. Now, what are these ACI, AQ, AT curves, and what are they depicting all these names? So first we'll go to the ACI curve. Now, ACI curve is the plot of photosynthetic rate against intercellular carbon dioxide concentration. It is nothing but a graph that gives us the rate of photosynthesis. Not the rate of photosynthesis, but the rate of carbon dioxide uptake, the net carbon dioxide uptake with varying levels of intercellular carbon dioxide concentration. Intercellular carbon dioxide concentration is nothing but what is the concentration of carbon dioxide inside the leaf. Now, if you look, we were talking about how a plant responds to environmental conditions. So, in that case, we should be requiring the atmospheric carbon dioxide concentration in this graph. But typically, this ACI curve is plotted in such a way that it gives us the photosynthetic uptake, or we call it assimilation, net assimilation with varying carbon dioxide, intercellular carbon dioxide concentration. Now, how do we know, or how do we get, how do we know what processes are responsible for this varying parts of the curve, or what are we going to get from this ACI curves? What is what we'll look into now. Now, if you see this curve, this is a typical curve that you can see for any for any C3 plant. You see that this is the concentration of carbon dioxide inside the leaf, 0, 200, 400, 600, 800. Now, as the carbon dioxide concentration increases from 0 to this high value of 800, this net carbon dioxide uptake is also going to keep on increasing. After a point, the rate of increase. Now, if you see here, the slope is high. That is, for every 100 micromoles of carbon dioxide, micromole fraction of carbon dioxide, we see that the net assimilation is increasing from 0 to a higher value. And as we move from towards higher values of carbon dioxide, the increase keeps on going up, rate of increase keeps on going up until one point when you see that even when the carbon dioxide concentration inside the leaf is increasing, the value of net assimilation is not increasing as much as it was doing in this part of the curve. So here there is a transition from a steep slope to a flat line, which we call it as the asymptote. Asymptote is nothing but this straight line where there is no change happening in the y-axis. So from this part of the curve, it is transitioning into another part of the curve where the curve, even if you keep on increasing the carbon dioxide concentration, there is no change in the net assimilation rate. Assimilation rate is going to remain at the same level. So this is how typically every C3 plant, the ACI curve for every C3 plant is going to look like. It is going to have an initial steep increase. Then at one point of the carbon dioxide value, that increase is not going to be very high. The rate of increase is going to be a little less. And at another point in the carbon dioxide concentration, you see that there will be no increase and there will be a steady line. A steady state is attained in the net assimilation. Now, what is the use of all this? I mean, what do we get by knowing the slope or transition or asymptote? So this part of the curve where there is a steep increase, or with every value of carbon dioxide concentration, when the net assimilation is increasing at a fast rate, or we can say the slope. We know that slope value means it is slope value is always y-axis, change in y-axis by change in x-axis. So if the value of the slope is high, means the curve is going to look, the curve is going to look something like this. Now, if the value of the slope is less, means the slope, it will be a very down, very flattened type of a curve. So we see that at this portion of at this portion of the slope, this slope portion or whatever slope value we are getting is giving us, or this part of the curve is giving us the maximum rate of carboxylation. Now, if you remember in the previous class, I had talked about three limiting factors that are going to affect photosynthesis. One is at what rate is the RUBP and carbon dioxide being combined together with the help of their Rubisco enzyme. So that is called the Rubisco carboxylation rate of photosynthesis. So that what is the maximum carboxylation that is happening is we can understand about the rate of carboxylation or the how fast the carbon dioxide is being fixed from this part of the curve. Now, this part of the curve, in where you find that the slope is transitioning into an asymptote, we see that we get information about how the RUBP regeneration. Now, in the third phase of the Calvin-Benson cycle is RUBP regeneration. The G3P molecule is going to get converted to 5-carbon RUBP. So, how fast is RUBP? What is the maximum rate of RUBP being regenerated? Information or the rate information regarding that rate of the process, that rate of RUBP regeneration can be obtained from this part of the curve. Similarly, if you see here, this part of the curve is giving us information on how is the G3P being utilized. Now, again, if you remember, we talked about how G3P is being converted to carbohydrate. So when it is getting converted to carbohydrate, it is one G3P molecule is going into the cytoplasm, and the cytoplasm is going to provide one phosphate molecule in return. So how fast is this process happening can be obtained from this part of the curve. So here we are getting the Rubisco limited phase. That is, if you see, the Rubisco enzyme is what is resulting in this carboxylation reaction or carbon dioxide fixation. So Rubisco limited phase means if the increase or decrease, so if the slope is high, we can say that the Rubisco is doing fast carboxylation. If the slope is very low, we can say that the carbon dioxide is being fixed at a very low rate. That is probably because Rubisco is not catalyzing that reaction sufficiently. So from this part of the curve, we can get how is Rubisco being a limiting factor to the photosynthetic process or assimilation rate. Does anyone have a doubt? Okay, anyone who switched on their mic, please switch it off. Now, in the next part of the curve, which is the RUBP regeneration limited phase, we see that how is the rate of RUBP regeneration going to affect the net assimilation rate. I hope everyone is familiar with the term assimilation. Photosynthesis, when you say photosynthesis, it is meaning how much of carbon dioxide is taken. Now, the net photosynthesis is when carbon dioxide is taken as well as carbon dioxide is released. Both these processes are happening. So when this A value is what is the net carbon dioxide being taken after all the uptake and the out, during photorespiration and dark respiration, some carbon dioxide is going to go out. So totally after all of these processes happen, what is the net uptake of carbon dioxide is what this A term is given. So this A term indicates net carbon dioxide uptake, which is also called as assimilation rate or net assimilation rate. So the x-axis is giving net total in the end of all these reactions, what is the net carbon dioxide that is being taken up by the plant. So this part of the curve curve indicates, or this part of the curve gives us information relating to the RUBP regeneration rate, and we can say that if the photosynthetic rate is around in this range, we can say that RUBP regeneration is what is impacting the photosynthetic rate. So we can say that if RUBP is regenerated at a very less rate, we can get the information from this part of the curve. Now, if it is RUBP regeneration is very high, also we can get the information about the RUBP regeneration rate from this part of the curve. Similarly, so we say this is the RUBP regeneration limited phase of the photosynthesis. So whatever rate of photosynthesis is happening in this part of the curve, we can say that it is being affected or controlled by the rate of RUBP being regenerated. Now, TPU rate is again triose phosphate utilization limited phase. So that means this part of the curve is showing us how fast or how slow the TPU is being utilized and how it is going to impact the net carbon dioxide uptake. Now, okay, we said that these three parts of the curves are what is giving us information regarding all these values of net photosynthesis. But what value are we going to get from knowing that this is a high slope or a low slope, knowing that this transition is not occurring very fast or the transition is occurring very quickly? What quantity is exactly being obtained from this type of the curve? So, like I said, the carboxylation limited rate is when reaction of carbon dioxide and RUBP happens. RUBP regeneration limited rate is when RUBP regeneration is controlling the net photosynthetic rate, and triose phosphate utilization is affecting the net. It is affecting the net carbon dioxide uptake or net assimilation rate. I just wanted to revise this triose phosphate utilization. In this triose phosphate utilization, for every 6 molecules of G3P produced, 5 goes for regenerating RUBP and only half a molecule is going for carbohydrate, which will move out from the chloroplast to the cytosol. And in exchange, one inorganic phosphate molecule is provided into the stroma. So this is the triose phosphate utilization just to understand that from the stroma, this triose phosphate is going to go here. In here, all the glucose manufacture is going to take place. In place of this triose phosphate, the cytosol is going to give back one phosphate molecule. So from here, we understood that the ACI curve is giving some information regarding Rubisco, some information regarding RUBP regeneration, and some information regarding the TPU utilization. When we say about Rubisco, we say that how fast or how strongly the Rubisco enzyme is going to help in the carboxylation process is what the Rubisco limited phase is. It is not giving us a quantity of Rubisco, but how is Rubisco enzyme going to make the is going to help in the carboxylation, whether it is going to increase the rate of carboxylation or decrease the rate of carboxylation can be obtained from this part of the curve. What is the rate of RUBP being regenerated and what is the rate at which TPU is being utilized, a triose phosphate is being utilized. Now that is ACI curve, which is the net assimilation rate versus intercellular carbon dioxide concentration. Now, the next curve that we talked about was called the AQ curve. Now, AQ curve is again the plot of net carbon dioxide uptake versus light absorbed by the plant or light absorbed by the leaf. Now, when the plant receives light from the sun, it is not going to be taken as energy of the sun. Instead, the photons are going to be absorbed, and that photons, how much photons are being absorbed by the leaf versus this net carbon dioxide uptake is what is this AQ curve giving us. Now, similar to the ACI curve, the AQ curve also gives us a lot of information about the photosynthetic rate. Now, if you see here, this curve is also somewhat similar to that AQ curve. You see this curve is again having an initial slope. That is, as light increases from the 0 micromoles of photons per meter square per second to 2500, we see carbon dioxide uptake is also going on increasing, and after a certain point, there won't be any increase. The net assimilation of carbon dioxide uptake is going to be constant. No change is going to happen. That will be the maximum rate of net photosynthesis. So even if you think that giving immense amount of light is going to help in the increase of photosynthesis, then no. Certain plants, it grows in the grows in indoor plants, and it will grow inside the house. Now we might think that, okay, let's go put it on the hot sun and it is going to get more light and it is going to grow big. Then no, after a certain amount of light, there is not going to be any increase in the photosynthetic rate. So this curve, a light response curve, is going to give us information about at what light intensity will be the maximum rate of net photosynthesis. Now, apart from that, it is also giving us another information, which is the rate of respiration. Now, here we see that the carbon dioxide uptake is negative. That mean, what does that mean? What do you think that means? When you say that carbon dioxide uptake is negative, means there is no carbon dioxide being taken up. Instead, carbon dioxide is being lost. So this part of the curve is giving us information about the rate of respiration, and that too not any respiration, but the dark respiration that is occurring. Now you see this part of the curve is giving us information about how much what is the rate of dark respiration that is occurring. So that is one important information that we are going to get from this AQ curve. Now, not only that, AQ curve, so we got two information. What is the light intensity at which the photosynthetic rate is going to be maximum? What is the rate of respiration? So rate of respiration in this case is going to be -4 micromoles of carbon dioxide per meter square per second. So that is going to be the rate of respiration. Apart from that, it is also giving us an important information about at what light intensity the carbon dioxide is not going to be taken in. I mean, the net carbon dioxide uptake, no photosynthesis is also occurring, no respiration is also occurring. So at this particular point, that is the light condensation point, is giving us the light intensity at which the net photosynthesis nor respiration is going to occur, but the carbon dioxide uptake is going to be 0. So that is also a very important information that we require when we are modeling a plant or modeling a leaf photosynthesis. And then again, light saturation point is nothing but the light intensity at which the rate of photosynthesis is going to be maximum. Beyond that light, how much ever light you give, the plant is not going to increase its photosynthetic rate. Now, the fourth important information that we are going to get from this curve is based on again the slope of the curve. Now, if you see here, the slope of this curve is higher than the slope of this curve. This curve is, I mean, the one with the dark color, the green color curve has got a higher slope when compared to the black color curve. Now, what does this, what information it gives us is, you see at the same light intensity over here, say it is around 200. So at 200 intensity of light, this black color curve, which might be the AQ curve for say some plant A, is lesser than the net carbon dioxide uptake of this curve, which might be representative of say some plant B. So plant A takes up only less carbon dioxide at 200 light intensity as compared to plant B, indicating that the light use efficiency, that is at a particular light, certain plants efficiently uses light to take up carbon dioxide in comparison to other plants. So in this case, from these two curves, what we are getting is the plant A, which is the black color curve, is less light use efficient. That is, it is not making use of light as efficiently it has to as compared to the plant B. So this information, that is this slope of the curve, is what, or we call it convexity term of the light response curve, is something. No, okay, no problem, Shreyas. Anyway, this lecture will be uploaded as video, so you can view it when your net connection is right, or in at whatever time is okay to you. Okay. So I was saying how the slope of the one another. Next information that we are going to get is the slope of the curve, and slope of the curve is giving us information about how efficiently light is being utilized for the net carbon dioxide uptake. So these four from the ACI curve, we are getting three rates. That is, three phases of the curve are giving us information on the rate of carboxylation, rate of RUBB regeneration, and rate at which triose phosphate is being utilized. And next, this AQ curve is giving us information about the maximum rate of photosynthesis, about at what light it is happening, at what light intensity is the plant, the carbon dioxide uptake in the plant equal to 0, and what is the rate of dark respiration. So in total, we got 8 informations, or 8 important components or parameters that is related to photosynthesis. Now, the next set of curves are called the AT curve, which is nothing but similar to how photosynthesis was measured for varying carbon dioxide and varying light intensity. Photosynthesis can also be plotted against varying temperature. Now, temperature is also going to affect photosynthesis. It is going to impact how far Rubisco, the catalysis of the carboxylation reaction. If the for different values of temperature, the enzyme will act in different ways. So AT curve is going to give us information about the optimal temperature. That is, at what temperature is the photosynthetic rate optimal? At what temperature will the photosynthetic not decrease, or what temperature will the photosynthetic will not keep on increasing? So from AT curves, we get this optimal temperature, which is an important parameter. So if you see in C4 curves, the optimal temperature is high, and not only optimal temperature is higher, but you see that the photosynthetic rate at that optimal temperature is also high. Now, if you come to C3 plants, you see the curve. This is all these are typical curves. So if you want to model any C3 plant, you can just refer to these curves to and understand what is the optimum temperature and what will be the photosynthetic rate. Now, there are models that very accurately predict these curves, but again, as a thumb rule or as a sort of reference, we can always use these curves to know whether the C3 plants have higher optimal temperature or C4 plants have higher optimal temperature or not. So we see that C3 plants are having the optimal temperature is not only is the optimal temperature less, but the photosynthetic rate at that optimal temperature is also less. And CAM plants, you can see, which is the gray light. So this optimal temperature is giving us information about okay, giving us information about how temperature is going to affect the photosynthetic rate. At high temperature, Rubisco deactivation will occur, as a result of which the carbon dioxide rate is going to decrease. So that is why beyond this point, you see that, see, if you see beyond this point, the carbon dioxide, if you keep on increasing the temperature, carbon dioxide is going to decrease. Now, that is why we say that the temperature should not be very high for photosynthesis for plants. So plants, it will function properly when the temperature is low until a point, but after a point, if you keep on increasing the temperature, what happens is it will negatively impact, that is, the photosynthetic rate will keep on decreasing. So this is what we get from the AT curve. Now you can see this is the T optimum we get over here for this particular from this particular curve, and we see that, say you're taking two types of plants, plants, or you're taking two species of C3 plants. We see that one has got an optimal temperature at uh, uh, at around 20. This will be the optimal temperature, and the maximum photosynthetic rate is this one. And for the other plant, you see the optimal temperature is this one, and you photosynthetic rate is this one. So you see, at all these rates, the photosynthesis will keep on increasing. But after this rate, the photosynthetic is going to become, it is going to decrease. It is going to reduce. Now, this part of the lecture is going to uh, put all of these in form of equations so that we can get one particular value for all of these parameters that we discussed. We discussed about ACI curves, we discussed about AQ curves, we discussed about AT curves. So AQ curves are going to give a certain set of information. ACI curves are going to give a certain set of information, AT curves. Now, what are all these information, and how we can solve these equations so that we finally will get the net photosynthetic rate. All this we are doing so that we can understand what is the photosynthetic rate of a particular leaf. So this model, the Farquhar von Karman-Berry model, it is a biochemical model that describes the rate of carbon dioxide assimilation in C3 plants. So what it is giving us is, it is giving equations that is linking the process of photosynthesis with the enzymes in the plant, with the light absorbed by the plant, with the amount of carbon dioxide that is available in the plant. It is linking all of this, and then they have developed a bunch of equation. And finally, if we solve, we will get the net photosynthetic rate. So again, this was introduced in 1980, and net photosynthetic rate. And this net photosynthetic rate is governed by these three biochemical processes: Rubisco limited phase, RUBP regeneration limited phase, and triose phosphate utilization limited phase. So you see, Rubisco limited phase is nothing but the carboxylation limited phase, when carbon dioxide assimilation is constrained by the activity of Rubisco. So if Rubisco doesn't work, it is going to reduce the carboxylation or the carbon dioxide from attaching itself to RUBP. So that rate is what, or uh, that information is what we'll get from this, or the rate of Rubisco limitation, or rate of photosynthesis at this phase is what we'll get uh, in the first phase of the ACI curve. So at low carbon dioxide concentration, when RUBP is sufficiently available, this will matter. If the Rubisco activity is less, even if there is a lot of carbon dioxide, it will not be able to fix the carbon dioxide. It is only with the help of Rubisco that RUBP can attach itself to carbon dioxide. So irrespective of how much carbon dioxide or how much RUBP is available, if the activity of Rubisco is not sufficient, photosynthesis or carbon dioxide fixation is not going to happen, which is an important process in photosynthesis. Now, it is represented by this notation AC in this all of these equations that we are going to study. Now, next is the RUBP regeneration limited phase. Now, you see it is also called the electron transport limited phase, and that is because during RUBP regeneration only, the ATP molecule is again being reduced to ADP. If you remember the Calvin-Benson cycle, when G3P is converted into RUBP, 3 ATP molecule is utilized, which releases 3 ADP. Now, ADP is again formed when for the production of ATP during the electron transport change in the photosystem 1, the electrons are again reducing one protein complex with the help of electrons. ADP is attaching itself to phosphate molecule and forming ATP. So this RUBP regeneration, if it is not occurring, or if the rate of RUBP regenerated is less, during that time ADP is being produced, and if that ADP is not produced, again the ATP during the light reaction is not going to happen. So you need to link the light reaction and the Calvin-Benzil cycle here. So maybe I'll take you back there. So if you see here in RUBP regeneration, ATP is being utilized to form ADP here. Now this ADP is what is being here, if you see in this photosystem 2, sorry, in the ATP synthesis in here, I'm sorry, during the photosystem 1, NADP is being reduced to NADPH. During the ATP synthesis, if ATP has to be regenerated, ADP molecule is required. So from where is this ADP molecule coming? It is coming when G3P is converted to RUBP, ATP utilized, ADP is produced. It again goes here, attaches itself to phosphate, forms ATP. ATP is again used here, which is again continuous in this cycle. So ultimately, if this G3P is not converted to RUBP or RUBP is not regenerated, what happens is this part of the reaction is affected. Now, it is also called the maximum electron transport rate, because it is this rate that is governing the proton gradient that is resulting in this ATP synthase to rotate. So you can see how intricately it is linked. So if this ADP has to be converted to ATP, this proton gradient should be there. This proton gradient is caused as a result of electron transport. Electron transport is what is resulting in this H plus molecules, H plus protons to come from here to here. So if that gradient is not there, this ATP synthase won't run. If ATP synthase won't run, ATP will not be formed. If ATP is not formed, RUBP regeneration will not occur. Now again, if you go the other way, if RUBP regeneration doesn't occur, ADP will not be supplied for ATP synthase. And again, that is also going to impact. So in here we say that we indicate this process of RUBP regeneration as also, or we call that process of RUBP regeneration as also electron transport rate, because those two processes are very much linked to each other. So this process is happening, or this phase is happening at moderate to high carbon dioxide concentration. Like if you see the ACI curve at moderate, if you see this part of the curve, here it is occurring at low carbon dioxide concentration. Here it is occurring at moderate part of the carbon dioxide concentration, and here it is occurring at high carbon dioxide concentration. So that is why we are saying this RUBP regeneration phase is occurring at moderate rate of the carbon dioxide concentration. So during this phase, Rubisco is saturated with carbon dioxide, and the limitation, that is Rubisco is no longer going to control the photosynthetic rate. Instead, RUBP regeneration will be what affecting the photosynthetic rate. Now, after this RUBP regeneration phase is over, it will shift to the TPU utilization limited phase, and that time how fast this TPU being utilized is what is going to impact the photosynthetic rate. So when assimilation is limited by the capacity to export and use triose phosphate, then this phase is going to govern how the photosynthetic rate is being affected. So Farquhar, von Kammerer and Berry model is using all these phases to estimate some parameters that will help us in obtaining the net photosynthetic rate. Now, the Farquhar von Kammerer Berry model is not just one equation. There are a lot of equations that is taking into consideration all of these processes. So we will go into how these processes are being represented, or what are the parameters that are used in defining this carboxylation rate. What is the parameter that is used for defining the RUBP regeneration? What parameter is used for defining TPU utilization? What parameter is used for the light compensation point for the maximum rate of photosynthesis, for light use efficiency? What are the terms that are used, or how are we going to get those values in order to in order to obtain the net photosynthetic rate of a leaf? Now see, if you see, the final equation is this one. A net is equal to minimum of AC, AJ, AP minus RD. RD is what? The dark respiration rate. AC is what? It is the Rubisco limited phase or Rubisco limited photosynthesis. AJ is what? It is the RUBP regeneration limited photosynthesis. AP is what? It is a triose phosphate utilization limited photosynthesis. So this is all rates of photosynthesis, except that AC is what is the the rate of photosynthesis over here is controlled by the activity of Rubisco. The rate of photosynthesis in AJ is controlled by RUBP regeneration. Rate of photosynthesis is controlled by the TPU triose phosphate utilization. So if you when we calculate, if say suppose we get the value of AC, AJ and AP. If we get AC, AJ and AP, then what is going to be the minimum value of all these three rates of photosynthesis? That is the value we'll take in this part of the equation. And then we will minus the dark respiration rate, which is the mitochondrial respiration rate. Now, why are we minusing it? Again, if you want to get the net assimilation rate, you have to know what is the total carbon dioxide that is being taken in and what is the total carbon dioxide that is being released out. So when you bring together these two, the input minus the output, you will get the net photosynthetic rate. Now, when I say the minimum of these three, we say that A net is equal to minimum of AC, AJ, and ATP or AP minus RD. What does this minimum mean? Now, if you see here, suppose there is a curve like this. Just a second, I'll. Say, suppose there is a curve like this. This is one ACI curve we are getting. This is A net and this is CI. This is one ACI curve that we are getting. Okay, this is the TPU phase. This is the regeneration. The middle part is the regeneration, RUBP, and the first part is the Rubisco limited. Similarly, we are getting another curve. Now, with one curve, we are not going to know what is the photosynthetic rate. So for that, we should have a number of curves for getting the photosynthetic rate. So first we'll get one curve. Second, again, we'll measure something and we'll get the curve something like this. And third, we'll get the curve something like this. Now, this is the Rubisco or AJ. This is the this is AC, this is AJ, and this is AP. Now this equation AN is saying that we have to get the minimum of AC, AJ and AP and then minus it with RD. So how do we get the minimum? In order to get the minimum, what we'll do is, in this part of the curve, what is the curve that is giving us the most minimum value? We'll take this one. This is what is giving us the most minimum value. In this part of the curve, again, this is what is giving the most minimum value. And or this part, this is the equation rate that is most minimum. And in this part of the curve, again, this is the equation. So the final ACI curve is going to be this one. So what is the AN related to, what is the net photosynthetic rate related to this? What is the net photosynthetic rate related to this? Say if this is slightly more, what is the net photosynthetic rate related to this? The minimum of these values is what will be taken as the value that is to be included over here. So say suppose we are doing that in terms of numbers. We have one column here. We have AC, AJ and AP. We got for first plant. So first curve we are getting one set, second curve, third curve. We are getting this as 3, this is 5 and this is 1. Here we are getting this as 2, this is 3 and this is 5. And here we are getting this as 8, this is 9 and this is 10. So in this case, the minimum of all these values. So if you see here, the minimum over here is this value, 2. Here AJ minimum is 3 and AP minimum is 1. So minimum of these 3 values is again 1. So minimum of these 3 is 2, 3 and 1. Out of this minimum again 1. So this value minus the RD value will give us the net assimilation rate. So this is just in very simplistic term, trying to indicate how we will be using the values. Again, when we go into the equation and solve all these, we'll be able to understand a little bit more. Okay. So this equation is the minimum value is what we say as the minimum of AC, AJ, AP. Whatever is the minimum value of the 3 values of net photosynthesis, we will add here. We will include in this term and then minus it with the dark respiration rate. So I hope this equation is, I think this is not very complicated to understand. You just have to find what is the minimum value of AC, what is the value of AC, AJ and AP. And out of these 3 values, which is the minimum photosynthetic rate, we will take that value and minus the dark respiration for the respective curve. Now, how is this AC value computed? How do we get the curve? And if you look at the curve, you can't just get the value. There is a range of values in that face of the curve. If you look at the response curve here, this curve is giving us information I told, but then what value is the photosynthetic rate in this Rubisco limited phase? The value is varying from say this some minus value until 20. So how do we get one particular value for this Rubisco limited phase? And that is given by this equation. In this equation, if you see, it is VC max into CI minus gamma star divided by Ci plus Kc into 1 plus 0 by K naught. Now, just for your information, you don't have to learn these equations by heart, but we should know what all parameters are coming in this equation. It is only if you understand what are the terms that are coming in that equation that we'll be able to understand the process that is responsible for giving us this rate. So here we see that there is one term called VC max. VC max is nothing but it is the maximum carboxylation rate of Rubisco. So bingo, there we were talking a lot about Rubisco limited phase. What is the rate of carboxylation? So this term VC max is giving us maximum carboxylation rate of Rubisco in the unit of micromoles of carbon dioxide per meter square per second. Now inside the bracket you see two terms, Ci minus gamma star. Ci is again from the ACI curve, you know, it is intercellular carbon dioxide concentration, intercellular leaf carbon dioxide concentration. That is in mole fraction, in micromoles per mole. This tau star or gamma star is called the carbon dioxide compensation point in the absence of mitochondrial respiration. So let's see what that is. Okay, before that, we'll go into these terms Kc, K naught, and O. Kc is something called the Michaelis-Menten constant for carbon dioxide, and K naught is called the Michaelis-Menten constant for oxygen. O is nothing but giving us the oxygen concentration inside the leaf. So we know what maximum carboxylation rate of Rubisco is. That is the rate at which the carboxylation carbon dioxide fixation is happening. We know what is the leaf concentration, intercellular carbon dioxide concentration. So we know what these two terms mean. We know what is O. O is nothing but what is the oxygen concentration inside the leaf. Now what we have to know are these three terms Kc, K naught, and tau star or gamma star, which is called the carbon dioxide compensation point. And these terms are called the Michaelis-Menten constant for carbon dioxide and Michaelis-Menten constant for oxygen. I think if I take all of this, it will be too overwhelming. So as of now, I will stop with this reaction, this final Farquhar FVCB. We call it FVCB in short form. So FVCB equation is giving us the net photosynthetic rate, which is the minimum of the Rubisco limited photosynthetic rate, RUBP regeneration limited photosynthetic rate, or TPO phosphate, triose phosphate utilization limited photosynthetic rate minus the dark respiration rate. So this is the final equation. How do we calculate the Rubisco limited photosynthesis, this AJ and AP? We'll look in more detail in the next class because I think there's just 10 more minutes and I don't think so I'll be able to cover the whole thing. So we'll go into that part in the next class. Before that, I think I'll launch one more quiz, which is which will like give an idea about what is the yeah. So I have launched one more quiz. This is just some basic questions on the ACI and AQ curves. So please do this as well. I think this will give you an idea about how far your understanding has been on ACI and AQ curves. And based on that, you can just if you have doubts, you can ask that. If you have doubts, you can post it in the Google form. Or if you want me to explain that once again, you can ask me to do that. So please do this quiz and then after that you can leave the session. Do whatever you know. I mean, I don't think I gave a lot of focus on the units of ACI and AQ curves. So if you don't know the units, no problem. Just write in what is ACI curve, what is AQ curve, what are the three major limiting factors for photosynthesis. And the last question, oh yeah, the last question also I talked about it. So you can just put in that information also. Those who have submitted, you can exit the session. I will be sharing the quiz results and in the WhatsApp group. Thank you for attending today's session. Mayank, did you attend the quiz? Mayank, if you haven't attended the quiz, I can relaunch it for you. Otherwise, I'll share it by email or Google or WhatsApp, and you can attend it there. Do you want me to relaunch it? Don't seem to be getting any response from you, so I will assume that you have you wouldn't like to attend it. So thank you for attending the session, Mayank. You can leave the room.